Okay, so last time we talked about microevolution and evolution within populations in generational time. Now we're going to talk about evolution on a large scale. And this is where we get into things like speciation. And so we're going to talk about those processes. Let's see if my computer is going to work here. Hello. Thank you. Okay. Objectives, as always, make sure that you can go through these. Most of these are really related to one another. And if you remember, we talked about homologous and analogous structures within our support for evolution. So keep that in mind. Let's just jump into it. This is some of those things that's kind of fun to understand once we get into it. If you know, I am a nerd. I am a huge nerd. I got really excited. I get really excited about a lot of stuff. I grew up at the time when Pokemon was new and fresh and exciting, and Eevee was my favorite Pokemon at that point in time. Now that I'm older, Slugma, primo Pokemon right there. I love me a Slug. But Eevee and the Eeveelutions are a great example of macroevolution. Okay. Now I know that in the game and in the show and things, Eevee is given a stone and it evolves directly into one of three: Vaporeon, Jolteon, or Flareon. But when we think about macroevolution, this is over a very long period of time and can result in speciation, where individuals split into one or more or two or more new species, and so they share a common ancestor. So arguably, if we think about it, Eevee is the common ancestor of Flareon, Jolteon, and Vaporeon. But when we look at the three different evolutions, we are not going to find them in the same environment. They are now adapted to completely different worlds. Flareon is not going to be found in the same place as Vaporeon, and so I am not likely to see any sort of reproductive event between those two species of evolutions. Okay? And this is where it gets into that macroevolution. When organisms become adapted to their environment, they may or may not choose to mate with other individuals that are not like themselves. And this ties into that sexual selection and that non-random mating. And there's a lot of different ways that people and scientists go about defining species. We're going to use a particular one in this class, but just keep in mind that there's a lot of different ways that this can happen. Okay? So, I love it because if we were to look at the macroevolution of dinosaurs, for instance, there is a common ancestry between dinosaurs and birds. My husband bought me a birds shirt, and they're all dinosaurs, and it's the dinosaurs within this Saurischia grouping. These Saurischia are what gave rise to our modern day birds. Birds are reptiles as a result. And you've probably heard that, like, oh, the T-Rex, its closest living relative is a chicken. Yes, that's a little bit of a simplification of what that is, okay? So let's talk about what makes a biological species so that we can better understand macroevolution, okay? If we talk about a species within the terms of the biological species concept, okay, this means that this is a group of individuals that are sharing a gene pool. They have alleles in common. They can interbreed with one another, and they are reproductively isolated from each other. Breeding with one another increases their fitness, but breeding with individuals outside of their gene pool, outside of their population, decreases fitness. Hybrids are not good. Individuals that are hybridized are not good. Now, this is not based on appearance, okay? For instance, these three birds are different birds. They're different bird species. I know, it's crazy. Flycatchers, the bane of any sort of bird watcher's existence, because these guys like to sit at the very tippy tops of trees. They all look the same. They move in the same way. You can't look at their behaviors to determine who's who. You have to hear their calls, and their calls are very, very different. In that way, that their calls are so different, even if they live in the same environment and they're in the same tree, they will not recognize each other as mates. And part of that is because if they hybridize, it decreases their reproductive success. And as we know, we all are biologically driven to have fitness and reproductive success, especially if we're talking about wild animals, okay? Humans don't count as far as the biological species concept. We can look very, very, very different, but because we can all breed with one another and we share a gene pool, humans are all one species in Homo sapiens, okay? we might have cultural and other differences that might deter individuals from reproducing with one another. Like these two individuals in this image would probably never even meet, and that's a global geographic distribution factor. But at the end of the day, they could make a baby, and so humans are all one species, whereas these flycatchers are not. Okay, so what do I mean when I say that individuals are reproductively isolated from one another? Well, we talk about these reproductive barriers, okay? These are isolating mechanisms that prevent hybridization. Hybrids reduce fitness. Hybrids may not survive. They may not be successful at their own reproductive attempts. They may not be able to raise offspring. Generally speaking, we don't want hybrids. Hybrids reduce the genetics of individuals, and a lot of times if we see hybridization happening, it can reduce individuals to basically extinction. One of the common examples of this is coyotes and red wolves. When there are enough red wolves in a population, they do not want to breed with coyotes. They do not want to breed with coyotes. They chase coyotes off. They actively kill them as a way of preventing them from being in the same territory. But red wolves are endangered, and there's about 30 individuals left in the wild right now. And if your choices are breeding with a coyote or not breeding, you're going to choose to breed. That's the driving force of a lot of these organisms. But those offspring now only inherit half the alleles from the red wolf. So they're only half red wolf. And if they don't have a choice to mate with a red wolf and they end up mating with a coyote, now their babies are only 25% red wolf, and so on and so forth. And it can result in genetic extinction of that species. And we've seen this occasionally within species, especially if like an invasive species is introduced. Mallard ducks are a good example of this. There was an American black duck that was hybridized so thoroughly with mallard ducks that there aren't any purebred American black ducks anymore, and they're functionally extinct at that point. Okay, hybrids reduce fitness and they reduce the genes of those individuals in the gene pool. So how do we prevent hybridization? Well, there are two different barrier types. You can have prezygotic and postzygotic barriers. Prezygotic barriers stop the baby from forming. Okay, no before baby, right? We are stopping this process in its tracks. Postzygotic barriers are after a sperm and egg have gone together, a zygote has formed, and this is where we start to see this happening. Okay, so there are 5 different prezygotic barriers and 3 different postzygotic barriers. Let's talk about them with pictures because pictures are helpful. Okay, so in our prezygotic barriers, we have temporal isolation. Temporal isolation is not referring to temperature, it's referring to time. Okay, in this instance, we have a striped skunk and a spotted skunk. Both skunks are found in the same environment. They eat the same sorts of foods. They have a lot of the same sorts of behaviors, but one becomes reproductively available and has that reproductive event occur early in the spring and the other one is later in the spring. It'd be like if a flower blooms in May and another flower blooms in December. They are not going to be used to fertilize one another to do that pollination event. And so that's what's happening with these skunks. While they might interact on a regular basis, they are not reproductively receptive at the same period of time. And so that results in this time isolation. Okay? We also have things like habitat isolation. If you are an animal and you do not live in the same place, the chances of you reproducing with an individual from outside of your environment are very, very low. Again, we break the rules as humans. We're global. We move around everywhere. You can get on a flight and go to China, tomorrow. That's cool. This snake cannot. In addition, if animals are utilizing different habitats, they probably have different behaviors. They probably have different favorite food sources. How are they going to raise their offspring? We're going to see that habitat isolation is a huge driving factor in these barriers. Okay? And then the last one that occurs before a mating attempt happens is behavioral isolation. And birds are a fantastic example of behavioral isolation. Birds have extreme behaviors to ensure that the two individuals who are having offspring are going to be successful in raising those offspring together. The example on this image is the blue-footed booby. Okay, there are tons of boobies, and it's fantastic. Bird people named all of their birds like kind of dirty names. There's a bird called a dick sizzle. There's a bird called a screamer. All right. Blue-footed boobies are doing, in this image, their foot dance. This is a mating dance that they do because blue-footed boobies live on the same island and breed at the same time as pink-footed boobies and brown boobies. And so if they are not matching up with the same individual with the same gene pool as them, there's a good chance that their offspring won't survive. It won't have good success. They maybe don't feed them quite the same things. There's other stuff that can happen here. And so behavioral isolation is another mechanism that's preventing any sort of zygote formation or reproductive attempt. Okay, so these three prezygotic barriers, your temporal, habitat, and behavioral isolation, are going to occur before in a mating attempt. Because again, hybrids reduce fitness. Now, these are not standalone. None of these are standalone, okay? Oftentimes, these are kind of fail-safes, and so if one fails, the next one kicks in and kind of stops things from going further. But if we bypass all three of these, if we bypass the temporal, habitat, and behavioral isolation, we can run into things like mechanical isolation. Mechanical isolation is when the parts don't fit. Okay, this is a great example here. So this is two different birds of paradise flowers that are both pollinated by the same pollinator. In the first example here, the flower has its stamen and carpels, its pollination parts sticking up. And so as that hummingbird goes in to feed, the nectaries are actually behind that pollinator part. And so the hummingbird gets pollen all on its chin. Okay. If that hummingbird doesn't immediately visit another flower of the same species and it goes to the other bird of paradise flower, that one, instead of having the pollen get on the chin, it has its pollen parts hanging down. So when the hummingbird comes in to feed on the nectary, the pollen goes on the head. So this bird is going to have pollen on its forehead and pollen on its chin. In this way, when it goes to visit the next flower, it doesn't matter which species it is, it's carrying the pollen of that matching species. This is that mechanical isolation. They are physically separated in where those parts are. Another fantastic example of this would be if it's not really true speciation because dogs are all one species, but if you tried to breed a Great Dane to a Chihuahua, you're gonna have some problems, okay? The parts aren't gonna fit. That's mechanical isolation. If we bypass all four of these and we move into gametic isolation, like these sea urchins do, it means that the sperm and eggs do not recognize each other, okay? So sea urchins are what we call spawners. They release all of their gametes into the water at the same time. And in this case, purple and red sea urchins are in the same environment. They spawn at the same time. There's no behaviors because they're just releasing their gametes into the water. There's no mechanical isolation because, again, releasing gametes into the water. So how do we end up stopping hybridization here? There are physical actual proteins on the egg and the sperm, and if the sperm were to encounter an egg of a different species, it would not even begin the process of attempting fertilization. So gametic isolation is the gametes don't talk to one another, okay? Again, all of these play a role together, and all of these are a way of preventing hybridization from occurring before a zygote is formed. But again, we can bypass these. Sometimes that happens. So what do we get when the prezygotic barriers fail? Well, there's three things that we generally see in what we call the post-zygotic barriers, or after a zygote has formed. Reduced hybrid viability, also known as increased mortality, okay? Reduced hybrid fertility, or infertility of hybrids, hybrid infertility, or hybrid breakdown, or F2 breakdown, or F2 fitness, okay? You'll see those terms kind of interchangeably. Now, with reduced hybrid viability, This is something we see with like amphibians, okay? Amphibians are also just a little bit loose with the whole preventing hybridization, okay? This is a newt species that is a hybrid of two particular different species. And what you might notice if you're looking is that his little tootsies aren't working properly, okay? His little tootsies are kind of turned a little bit, and that's going to affect his ability to run and move effectively from predators or to find and hunt for food. We know that if you are eaten, no babies being made, or if you don't get food, no babies are being made. Other great examples of reduced hybrid viability is when humans try to make hybrids in captivity because we think they're cute. There have been attempts to cross different big cats together, like pumas or mountain lions and ocelots. And while they can successfully reproduce, the offspring cannot survive because neither mother can produce milk that is appropriate for those babies. So the babies die. Okay, it's not good to do that then. We've reduced the viability, we have increased the potential of hybrids dying. And again, that's reducing fitness of those populations. So we don't want that. Okay. Reduced hybrid fertility or that sterility is probably the most classic example. If you started thinking about hybrids, your first thought was probably like a mule or a liger. Okay, mules and ligers are generally going to have reduced fertility, and a lot of that actually has to do with chromosome numbers. So horses and donkeys have one pair of chromosomes different. And so mules actually have a 2n+1 situation, which is going to then affect their fertility, okay? Mules, sometimes you'll get a mule who's fertile, but that's going to be a non-disjunction event. Ligers too, same sort of thing. They generally cannot reproduce. And again, if I am trying to pass my genes on to future generations in my fitness and my offspring can't reproduce, that defeats the purpose. Okay, and then the last but not least kind of post-zygotic barrier we see is the hybrid breakdown. I've personally experienced this, especially plants do this a lot. Plants, when they're hybridized, can often produce really large, robust plants in that first hybrid cross, where you took the two purebred plants and crossed them. We did this when we grew peppers here in North Carolina. We had pepper plants that were a Dragon Cayenne, and so it was a pepper hybrid. That first generation, those plants were taller than me. They were like 6 feet tall. They looked like small trees, and I was up to my eyeballs in peppers. I had so many pepper fruits. And some of the peppers fell into my garden, and I just let them go the next year. And so the next year I go out, and the plants were viable in that they started growing. But that second generation from that hybrid, that F2 fitness, that F2 hybrid, grew maybe about a foot tall and maybe had 10 peppers on it. Not as good as the first one that was over 5 feet tall and looked like a tree, okay? When I let those peppers go again the next year, because I was just curious and I had been a teacher at this time, they barely grew and I almost didn't know they were peppers. I almost pulled them out of my garden as weeds. This is something we see a lot in agriculture because hybrids are going to be more robust and they will produce a lot of crop, but you cannot use the seeds from hybrids to successfully grow crop the next year. And we have seen a lot of small farms actually go out of business because they can't afford to maintain the prices of the hybrid crops, and the seeds don't grow. So hybrid breakdown does happen, and it's going to be over time and generations. And again, it's just decreasing fitness, right? If we're decreasing fitness, we're going to want to avoid that as a species. Okay, so those are your pre-zygotic and post-zygotic barriers. Do make sure you know them and do make sure you can give examples for each. Okay, we are going to move on to some of the models of speciation here then. What really comes into the driving force behind some of these different sorts of barriers that exist are going to be how individuals are interacting with one another in their environments. So there are two big ways that speciation can occur: allopatric or sympatric speciation. Okay, patric means land, allo means different, and sim means same. So oftentimes, and this one makes sense to most people, if I have geographic isolation for really long periods of time, in these two different environments, and these organisms are adapting and evolving within these two environments, if they were to come back together, they're not able to interbreed. Those individuals would be considered two different species. This would be like your marine iguanas and your tree iguanas. You will not find those two making babies together at any point in time, even if they were to come back together again. Okay, so that's going to be your allopatric speciation. Sympatric speciation is a little bit different. And we often see allopatric speciation, such as a new species growing on an island, right? Like they got moved out to an island through the founder's effect. And so they're now separate from the original species, right? So we have allopatric speciation where the island species is now different. But if that island species becomes reproductively isolated into groups, we might see then that sympatric speciation. This is something we see with like the cichlid populations in Lake Victoria, which is a large landlocked lake in Africa. The cichlids are going to be utilizing different parts of the lake, different resources, and it becomes almost allopatric speciation, even though they're in the same geographic area. Because if I am a species that lives on the surface, I'm never going to encounter a species that lives at the bottom of a very, very large deep lake. Okay, but at the same time, I'm in the same geographic area. Another great example of this, though, is going to be in things like our Galapagos finches, where we do have what's called adaptive radiation. Okay, and this is going to be where new species evolve from a single ancestral species. So again, allopatric speciation, founder's effect to a new island, sympatric speciation, adaptive radiation follows. And so these populations became adapted to the particular habitats. And so now, even though these guys share a common ancestor, I'm not going to find the large insectivorous tree finch trying to make babies with the warbler finch. They have two completely different beaks. They are eating completely different foods. They are in completely different environments. Even though if they're on the same island, they are now two completely different species. And we see that as an accumulation of those different prezygotic and postzygotic barriers accumulating over time. Okay? After adaptive radiation, we do need to consider what happens when a species fails to adapt. Okay? Mass extinctions or extinction events in general are a very normal part of a species process. If a species doesn't adapt, if we don't see natural selection, if we don't see evolution, if we don't see a way for these species to overcome any of the challenges, they may and probably will go extinct. So I had mentioned like the coyotes and the red wolves. And the American black duck and mallards. We are seeing some of these rare species going functionally extinct because their genes are getting so diluted in the gene pool that they're no longer any purebred individuals in the wild. And that's going to be what we see happening with more and more invasive species if we see hybridization occurring with some of the shifts and changes in geographic regions. What we thought might have been allopatric speciation, we're finding maybe it wasn't. There's a lot of things happening there. We do also know from the fossil record that there have been at least 5 mass extinction events that have occurred in life's history. Again, probably way more because as you remember from chapter 14, fossils are not complete. The fossil record is not complete. And so we don't have a full picture. But we do know that extinctions are when we're going to have really high percentages of individuals going extinct, with in some cases estimates being up to about 95% of species, such as what happened during the Great Oxygenation. We know that, for instance, the end Cretaceous period is when the meteor hit the Earth. We know where the meteor hit. There is evidence of it. And you can imagine that if a 6-mile wide meteor hits the Earth, it's going to vaporize everything and change the atmosphere. And so as a result of that atmospheric change, we knew there was a mass extinction with those dinosaurs, with those cold-blooded organisms, and your mammals took over. Okay, so mass extinctions are absolutely going to happen. If species do not adapt to their environment, they will become extinct. If species cannot handle reproductive success, they'll go extinct. If we have hybridization events, extinction will occur. All species end in extinction just like life ends in death. Okay, so check your understanding quick, and then I'm going to switch gears to a little bit more information that'll set us up for Unit 5. Okay, so Unit 5 is our diversity of life, and it's going to be a crash course survey of the living world, which is honestly a lot because it's a crash course survey. So we're going to go through microbiology, mycology, botany, and zoology, and it's fun, okay? It's going to be fun. You're going to have tables to fill out. It's great. So basically what this means is we have 3 domains, okay? Looking back through evolutionary history, we know that we have bacteria, which likely arose first, and they are prokaryotic cells. And then after that, we have archaea, which are also prokaryotic cells. And then finally, eukarya, which are eukaryotic cells. We've been focusing on this entire semester with those cells that have those large organelles, the nucleus, and that sort of thing. And eukarya includes the kingdoms protists, plants, fungi, and animals. So we're going to survey both bacteria and archaea, and then all 4 kingdoms of the eukaryotic world in Unit 5. In order to fully understand that and to better be prepared for Unit 5, we need to talk a little bit about taxonomy. Okay? In science, taxonomy is the act of identifying, naming, and classifying organisms. We do use the Linnaean binomial system, which was developed in the 1600s, and it's a genus and species specific epithet. So that means that if I am talking about humans, I am talking about the species Homo sapiens. If I am talking about the American toad, I am talking about Bufo americanus. When we name things with the binomial naming system, okay, the genus is capitalized, the species is lowercase, the whole thing is italics, and you can abbreviate the genus. So we could say H. sapiens, and that refers to humans. But we generally say it once full out, and then we would abbreviate. Taxonomy is a hierarchical nested system. And so when we talk about domain eukarya, that includes all organisms that are eukaryotic. If I talk about kingdom Animalia, I'm only talking about animals. Phylum Chordata means animals with a backbone of sorts, okay? Class Mammalia, mammals. Order Primates. Family Hominidae. Genus Homo species. So this is a way for us as scientists to put things into nice little boxes so that if I'm talking about something, everybody knows what I'm talking about, okay? Does anyone happen to know what Vulpes vulpes means? I told you Catus felis is the house cat, and Bufo americanus is the American toad. Vulpes vulpes is the red fox. It means fox in Latin. Okay? When we look at taxonomy and we have an understanding of species, we actually can start looking at ancestry as well. And so we want to look for common ancestors, and there's a couple ways to look for that. One, we can use phylogenetic trees. Phylogenetic trees are going to nestle in those common ancestors, and everybody that comes after that classification is going to have traits that are shared with one another. So sheep, cattle, red deer, and reindeer are all related. They are all in the order Artiodactyla because their feet are the same. They have those cloven hooves, those split hooves, and they actually have them in 4. And so if you look at deer prints or cow prints, they look different based on size, but the physical structure is the same. So these guys are all classified as Artiodactyla because they share a common ancestor that had that foot shape. Okay? We then split out into the Bovidae and Cervidae families, or the bovine or deer families. In this case, we're seeing huge differences here, mainly in the head ornamentation. Sheep and cows have horns. Deer have antlers. Horns grow continually through life. Deer shed their antlers every year. And so that's going to be another big division, and it's going to be something that is shared between these organisms here. Okay? Sheep are obviously different than cows. I think we all understand that. And there's going to be differences in the wool, the size, the reproduction, that sort of thing. And so we have 2 different species within 2 different genus. But if I were looking at this dairy cow here, or if I were looking at a meat cow, oh gosh, I'm going to make myself, like an Angus cow, like black Angus cow, they might be different species, but they are of the same genus. They're still Bos, okay? And then your deer, your red deer and your reindeer, 2 different genuses, but they are still deer. We're still not going to see them reproducing with one another because they are different species, but they share a common ancestor in being deer, okay? Another way we can look at this is by looking at what's called a cladogram. Cladograms in phylogenetic trees are pretty interchangeable. A lot of times we use them. They're technically different. It's fine. Okay. So what I like cladograms because cladograms actually allow us to put the particular traits we're looking at on the phylogenetic tree that we are building. So we can look at cladograms and we can determine a series of traits that we're interested in of individual organisms. And then we basically say yes or no, right? Yes or no. Do they have these traits or do they not? So if I look at the traits I have here and I'm looking at a lancelet, which is a small invertebrate chordate, an eel, a newt, a snake, and a lizard. Okay, they all have a notochord. Cool. I'm not going to put that on here because that's going to be an early trait that was shared by a common ancestor. But if I look at my next thing, vertebrae, lancelets don't have vertebrae. So they become what's called the out group, or my first group that's the most ancestral, the oldest group. Okay, they're not going to have any of the other traits that I'm interested in. And so then I go to the next one, or lungs and a 3-chambered heart. That drops the eel out. Internal fertilization and amniotic membrane. That drops out the newt. And then the other ones, I don't have any real consensus here. So 4 bony limbs and a long cylindrical body are not going to be good traits to use in determining evolutionary relationships. And this is because we're looking again at those homologous traits, things they share in common because of a common ancestor, or analogous traits like having 4 bony limbs, right? That's kind of analogous. Now, we know snakes have vestigial limbs, and so we could argue that they have 4 bony limbs, but that is going to be something that, it can get a little confusing then. It's easier to lose a trait than it is to gain a trait. And so we're not generally looking at lost traits or vestigial traits here. We're looking at things that are still common and able to be shared. So when I read this cladogram, okay, the first one out, my lancelet, is my oldest species. It is not going to have any of the other traits on this tree. It's only going to have the trait that we determined is ancestral, in this case, the notochord on the embryo. If I look at my snake and lizard, we have an amniotic egg and internal fertilization. That's what separates them from the newt or the eel or the lancelet. But snakes and lizards also have lungs and a 3-chambered heart and vertebrae and a notochord. So my organisms at the most end of my cladogram are going to have all of the traits that I'm interested in, and then I'm going to see those divisions or branches occurring when the traits are found that are no longer in that particular species. I will ask you to read a cladogram, also known as a phylogenetic tree. Either way of calling it is fine. I will ask you to read this, so do practice doing that. I know that the pre-lecture work for this one is about a cladogram, so do watch that and do make sure that you understand that. Here's your Check Your Understanding for the second part of 16, and that's it. If you have any questions, let me know. Start studying for your test. Thank you.